Hi there, Year 12. In the video for Year 1 or AS Biology, looking at how things cross the membrane. So today we'll be looking at diffusion, facilitated diffusion and active transport, once I've had a cup of tea. So let's have a look at it then. So start off with diffusion, or what will often be called simple diffusion. I would always recommend putting simple diffusion in the exam rather than just diffusion, because often they will accept diffusion, but sometimes they won't if they want you to be more specific. So, what is diffusion? Diffusion is the net movement of particles or the net movement of molecules or or ions from a region of higher concentration to a region of lower concentration. So even my definition on here isn't very good because you should use comparative language. That should be higher with an ER on the end and lower with an ER on the end. The factors that affect rate of diffusion in the natural air around us, if it was just diffusion of particles, would be the steepness of the concentration gradient, the temperature, surface area, and properties of the diffusing substances. But we're going to look particularly at diffusion over a membrane. So, what things can then just do simple diffusion across a phospholipid bilayer? Well, the things that can simply diffuse are going to be your smaller non-polar particles, and they include oxygen because it's very small and non-polar, carbon dioxide, which is polar but is small enough to fit through the phospholipid bilayer, water because it's a polar molecule, but it is small enough again, and lipids because they are fat soluble, and fat soluble molecules can sort of fuse the membrane to allow that to happen. Now, just be really clear with this. When we're talking about simple diffusion, it is across the phospholipid bilayer. Never say across the membrane, because the membrane could mean through the proteins and things like this. And in this case, we're not talking about proteins. We're talking about the molecules that are small enough to fit through the tiny gaps and are not repulsed by the hydrophobic layer between the phospholipid bilayer. So oxygen, carbon dioxide, water, and lipids are able to do simple diffusion through the phospholipid bilayer. There are two main protein types, however, as you should already know, in the membrane structure. They are the channel protein, which is here at the top, and basically it's a pore which allows the water to go through, or whatever molecule to go through this hydrophilic area safely. So I think of it like a channel, like a canal. And then you've got the carrier protein, and basically the carrier protein, the molecule which attaches in the carrier protein will be the specific shape to the carrier protein. That then fits into the specific shape of the carrier protein. The carrier protein then changes shape, and that allows the molecule to move through the membrane. So that, and it's definitely through the membrane because it's going through the proteins to get through the phospholipid bilayer. And it causes this movement of the carrier protein. So you have this sort of movement like that. Sometimes that's called the flip-flop mechanism. Sometimes it's called the ping-pong mechanism. But it's a change of shape you need to know. Now, molecules that can't fit through the phospholipid bilayer then are bonds that are either too big, which could be glucose. Think about the size of glucose, it's C6H12O6, or amino acids, which with the right R group can be a very big molecule. It could be charged molecules, such as ions, and also these molecules that are charged are unable to get through because they'll be repelled by the hydrophobic layer between the membrane, between the in the tails of the phospholipids. So they have to use channel proteins. The key thing is this is helping the diffusion across. It's not using energy. It's still moving from a higher to a lower concentration gradient. It's going down a concentration gradient, so it's still passive. So we call this facilitated diffusion because it's helping you do the diffusion. So what is facilitated diffusion then? So it's the idea that molecules still move from a higher to a lower concentration, but it is using proteins to help. And as I said, things with charges that are generally small enough that have a charge can go through, and there's a lot of ions through a channel protein, whereas carrier proteins tend to deal with things that are much more bigger. As I said, they attach into the carrier protein changes shape, and it allows the molecule to go through. Now, just to re-emphasize before we move on, that does not use energy if it's going from a higher to a lower concentration. The change in shape of the protein is a natural one that doesn't need energy to do it. So, simple diffusion then versus facilitated diffusion. What affects the rate of diffusion? Quite a common question where they might give you graphs in the exam. The simple diffusion is affected by the concentration gradient, as so is obviously facilitated diffusion. The higher to lower will have a big effect on the rate of diffusion. It is also about the thickness of the exchange surface in simple diffusion. So because it's diffusing across the phospholipid bilayer, if the phospholipid bilayer is a thicker layer, it's going to take longer to get across that point than if it's just a thin layer. And the surface area of the exchange site, obviously, so if the membrane's got a big surface area like the alveoli, that is going to give you a faster rate of simple diffusion. Facilitated diffusion, though, because it relies on channel proteins or carrier proteins, it will simply rely on the amount of channel proteins and carrier proteins that there are. So if you've got, let's say, a high concentration gradient, and you want to look at what's the rate of reaction, well, what will happen is, so I've just done that the wrong way around. So if you imagine that on the bottom of this graph, you've got a concentration gradient, what will happen with the rate of diffusion is it will increase for a certain amount of time of facilitated diffusion until you hit the point where there's no more channel proteins or carrier proteins available, and then it will level off because you can't push any more through those effectively those proteins. So sometimes, however, we go against the concentration gradient. So to go against the concentration gradient, we will need to use active transport. So active transport, an example of this is in the root hair cell shown here. And in the root hair cell, which absorbs minerals from the soil, such as magnesium, there's less minerals in the soil than there are in the actual root hair cell. So we have to go from a low concentration to a high concentration. But that's not just going to happen across the phospholipid bilayer. It's not going to happen for a channel protein because it would just move the opposite direction. So we have specialized carrier proteins in the membrane to allow that to happen. So active transport is when energy is required. The word active, obviously, it's against the concentration gradient and it moves things from a lower to a higher concentration. Now, there are some examples of this where we use something called coupled transport or co-transporter proteins, which we'll talk about to finish off looking at glucose as the example. So the carrier protein, as I said, just works the same way. It's just active transport there because it's going against the concentration gradient. So the shape of the active transport protein changes. That requires energy to do so, and that moves the material from one side to the other. Now, the difference is with active transport, it always works in one direction. So when we talked about facilitate diffusion earlier, the transport proteins carry it in both directions. In this case now, it's designed to carry it, for example, into the root hair cells only. So examples, we're going to leave that one. Examples that we need to know then, also it'd be worth you just being aware of, there are something called bulk transport. So when things move into the cell, and a lot of them, it's called endocytosis. And basically what might happen is the cell membrane might like infold inwards, allowing the molecules in, and you'll have come across exocytosis before, which is where materials are moved out of the cell. Two other terms which you will be worth you knowing as well is phagocytosis, is cell feeding, when it takes up solid materials, and pinocytosis is when it takes in liquid. Now we've done about the factors affecting the rate of facilitated diffusion and simple diffusion. I've just realised that I've rewrote simple diffusion there, so I'm going to cross that out. The factors affecting the rate of active transport then are the following. Okay, so the speed of the individual carrier proteins, if they work faster, obviously things are going to be transported quicker across the membrane. The number of proteins, but also the rate of cellular respiration. Don't forget, active transport uses energy, therefore it needs ATP. And therefore it needs respiration to happen. So the rate of cellular respiration will affect the rate of active transport in a cell. Just a picture of exocytosis there showing you how it removes materials, and then endocytosis being the opposite, the enfolding of the membrane to do so. So last bit then is co-transporter proteins. So this is something I always really try and push because it comes up in loads of different areas of the module. It's really important to learn. You come across it again in digestion and that. Now, I will just say this is a model of co-transporter proteins. They will work slightly different ways. So you might see slightly different examples in your textbook, but you do just need to try and make sure you know them. Basically, the type of carrier protein which binds two molecules at a time, they might take one molecule against the concentration gradient or another molecule with the concentration, so something called symport, uniport, lots of different examples, but energy is used. So we'll look at this together using the glucose in the gut as an example. So what we've got here is glucose, and it is being moved from the small intestines, or the ileum, okay, of the small intestines into the blood here. And how this works is via a co-transporter system. Okay, so we're going to look at this and actually make sure we understand the stages required to move that glucose into the blood. So let's start with a bit better picture here, which will help us understand a bit more. So just to remind you then, we have got the intestines here, the ileum of the intestines, which is part of the small intestines, and the blood. In between, we've got the epithelial cells, which are the layer of cells where the exchange happens over. The first thing that we need to do is we need to somehow get that glucose, which has been digested, into the bloodstream and across the bloodstream. So we want it to move across here. But the glucose won't move in via simple diffusion, it's too big. Okay. It also has got more, we've got more glucose now in our blood. So the first glucose will move by facilitated diffusion, but then we end up with more glucose in our blood over here and less glucose here. So we've got to move it across via active transport. Now, how do we do this? Well, this is an active process overall, but it involves bits of active transport and facilitated diffusion. So let's go through. So if we look at the red bits represent sodium. So if we look at just inside the epithelial cell to start off with, inside the epithelial cell is lots of sodium. So we use energy to pump sodium out of the epithelial cell into the intestines. Okay, that uses energy, so that is your active part. That does actually cause some potassium to move in, because it's what we call a sodium potassium port, so it's called a symport process. Okay, but all we're looking at is the sodium. So now we'll have lots of sodium in the bloodstream. We've moved all these sodiums out of the epithelial cell. So the sodium in the epithelial cell is low. This means the sodium in the the sodium in the intestines is now higher than the sodium in the epithelial cell. So that wants to then diffuse through into the epithelial cell, because it wants to move from a higher concentration in the intestines to a lower concentration in the epithelial cell. The only way sodium is allowed through the membrane, however, is to go through something called a co-transporter protein. So sodium will only be allowed through if it brings one glucose molecule with it as well. So every 2 sodiums brings 1 glucose molecule. So every time the sodium gets brought in, it brings a glucose molecule with it too. So you end up with a buildup of glucose molecules in the epithelial cells. Okay. Now, if you imagine the green is the glucose molecules I'm drawing here, okay, you end up with lots of glucose molecules. They then want to transport out of the epithelial cell. Well, they can't go back into the small intestines because there's no way of doing that. So they travel through a carrier protein, which would allow them out into the bloodstream as a result. So let's go through it stage by stage. Step 1, sodium is actively transported from the ileum cells into the bloodstream, so the epithelial cells into the bloodstream, using a sodium potassium pump. This uses ATP, it uses energy. So they would need to have mitochondria in those cells to allow that to happen. This means there's a lower concentration of sodium in the cells compared to the lumen of the small intestines, or the ileum. As a result, sodium then moves down the diffusion gradient from a higher concentration in the lumen to a lower concentration in the epithelial cells via a co-transporter protein. The co-transporter protein carries a glucose molecules into the cell with every 2 sodium, and that therefore increases the concentration of glucose in the cells. The glucose then diffuses out of the cell via facilitated diffusion, and then it goes into the bloodstream. So I hope that's been useful for you guys, and if you are a bit struggling about that, we'll look at that again when we do digestion in the future, but really important to learn that co-transport process with glucose. Thank you.